All right. So this was the last slide of our last PowerPoint. Part 3, I think. We're at the halfway point of this chapter. But this picture is from the Nobel Prize website, which I just opened up a minute ago, and we're going to go to again. So it's great because, you know, as most of you guys wrote in your comments about this website, right, the little cute animated things help, right, in remembering things. And so remember, too, as you're doing that bonus assignment I told you guys about last time, you definitely need to draw to get those bonus points. And again, it's not a drawing contest, right? It's about active learning, right? That's what it's about. So here, they don't go into as much detail, right, as we've gone into about these interactions, specifically the interaction between these two cells. Now, of course, these cells are labeled, right? This cute little guy is a dendritic cell, right? And so he does have the ability to engulf stuff by phagocytosis, so that's what he's doing, right? He is a phagocyte, although not one of our main phagocytic cells. You really wouldn't classify him as a phagocyte. His more important duty is what he's doing here. And what would we call this process, what he's doing right here in this picture? Antigen presentation. Oh, look, the title of this drawing. and what we talked about last time, right? We focused in on this part right here, right? How it was processed. Okay? But we can kind of, you know, fill in the gaps here. So let's do that. So this is a dendritic cell. So what's not shown here is how he's presenting this antigen, this piece of the bad guy over here, right? That piece right there. That's the antigen, right? How is it that he's achieving that? He's presenting it using a molecular receptor that he puts on his surface, right? What do we call that? What grabs onto the antigen and presents it on the surface? Well, yeah, but it has a specific name, this receptor. So there's a clue right here. It's an MHC. But remember, there's two classes of MHC, right? There's two classes that will pick up antigens and display them on the surface of a cell. For a dendritic cell, which is a special type of cell, right? How many people say it's MHC class 1? I'm only raising my hand to demonstrate that's what I want you to do. Well, good. How many people think it's MHC class 2? Yay, yes, you won, right? It's 2. And because this guy we would call an APC, and what do we mean when we say APC? What does that stand for? Antigen presenting cell, right? So what cell has MHC class 2? Antigen presenting cell. Specifically in this drawing, it is a dendritic cell. Who's our other antigen presenting cells that we're going to talk about again today? So dendritic cell is here. What are the other two? Yep, B cell. Macrophage. Good job, Asia. Okay. So what is MHC doing? Yeah, it's presenting antigen. It is presenting that antigen. Yes. So that's who this guy is, right? They have him labeled helper T cell, right? But there's a restriction here, right? Only antigen presenting cells are the only ones that can talk to T cells. And it's because they have this co-receptor, specifically helper T cells, I should say. Which co-receptor do they have? CD4 or CD8? 4, right? CD4 is your answer here. What cell has this? Who is this? The helper T cell. What does it do? What does the CD4 do? Do I need to rewind? Is this one 4 or 2? 1. Here, we'll go all the way back to 4. So find the CD4 co-receptor. What is it doing in that picture? CD4 co-receptor. It actually binds to MHC class 2. Remember, this is their secret handshake. These are the only 2 cells that talk to each other. Antigen-presenting cells and CD4 helper T cells. This is their club. Or maybe cytotoxic. Oh, cytokines. She's trying to, yeah, cytokines. It's the one that talks a lot, right? The helper T cell talks to all the other cells, so we're going to keep talking about that help that it does. Maybe, is that what she's focusing in on? Help? Yeah. Okay, good. Cytokines. Good. No, that's good. Okay. Okay, so for today's lecture, we're on part 4. I was looking over it earlier, so I was at the, yep, 4, it's up, right? It's there? Okay. All right, so on part, I should probably add that to my front slide, right? Okay, so we've already looked at Mr. Dendritic cell, right? Who are the other 2 antigen-presenting cells we just mentioned? B cell, macrophages. I really messed up my animation on that one, huh? I don't know how that happened. Here. Oh, I don't know what I did wrong. Up here? I don't want it on that. I want you to go bye-bye. There we go. There we go. 1, 2, 3. Okay, so remember, these launch our adaptive immune response. Right? These guys are paramount to having that type of response. We've got to start out with antigen being presented by one of these professionals, right? Because can other cells in our body present antigen? Yeah, right? Via MHC class 1 or 2. How many people say 1? Who has MHC class 2? Just these guys, right? These professional antigen presenting cells. Can like viruses get inside other cells in your body? Oh yeah, we know this all too well, don't we? I don't know anybody who's not been sick with a virus, right? It's been living in a bubble if you haven't. They get inside of our cells. Do we present those antigens sometimes when they get inside of our cells outside to alert the immune system that there's a problem inside? We do. And we do that via MHC class 1. Right? Remember 1 as being the most predominant, right? Probably on everything, right? All nucleated cells. Even these guys, as we saw last time, present on MHC class 1. And they can do it even without getting infected by a virus, right? Because again, we need to get this information out there in such a way that we can respond. Right? If your friend tells you that somebody's a bad guy, you might believe them, right? If a police officer tells you that person's a bad guy, you're more apt to believe them. Why? He should know this kind of stuff, right? He's in a position in our society that a policeman should know these types of things, correct? In your immune system, the ones that really know about the bad guys are these antigen-presenting cells, right? So these are trusted leaders of our immune system, leaders of this adaptive immune response. Does that make sense to you guys? Okay, so now we're going to talk about these antigen-presenting cells one by one. So as you can see on this list, they can present via MHC class 1 and 2, as I mentioned. But these guys, dendritic cells, macrophages, and B cells, are the only ones that have MHC class 2. Okay? They're the only ones that can communicate with helper T cells. And why they communicate with helper T cells and how they do that is going to vary depending on what's going on, right, at the time in the body. One important distinction here is, notice dendritic cells, man, they're big talkers, right? They'll talk to naive CD4 and CD8 T cells, and in fact, for the most part, they're the ones that talk to those naive B cells and T cells that have never seen antigen before. What did I say? I said B cell, didn't I? I meant to say helper T cells and cytotoxic T cells, right? CD8 and CD4 T cells. I caught myself. So they've never seen it before. So this dendritic cell is going to help activate them. And again, this is a trusted communication here. Once we have that first exposure, remember that first exposure is going to lead to the production of memory cells. So if it gets in again and the dendritic cell recognizes it again, and if we have memory cells, it can interact with those memory cells and start the process all over again. During the process, right, naive are going to go and differentiate and proliferate into effector cells that actually go out and do stuff, right? And memory cells that are going to stick around in case we see it again. So depending on whether we start here, we always have to start here at some point with an antigen, right? We've got to activate naive T cells, whether it be cytotoxic or helper or both. The next time around, right, we may interact with memory cells. But the same progression is going to happen. Those memory cells, when they're activated, we're going to get effector cells and more memory cells. And as the infection is progressing, or as we're dealing with the situation, there's continued communication between the dendritic cells and those effector T cells as they're doing their job. So as I said, they're big talkers, right? They're talking to everybody at every point. Macrophages, on the other hand, are more accessory. They can notice the footnote, right? Only under certain instances are they going to actually interact and activate naive T cells, helper T cells at that. I mean cytotoxic T cells. Cytotoxic 8. Cytotoxic. They will interact with effector T cells and memory T cells. And notice that there's only one interaction here for helper cells, for B cells. They only interact with helper T cells. And notice that they're effector helper T cells. So these are, for the most part, a lot of times ones that have already been activated by a dendritic cell. So why is a B cell communicating with a helper T cell? Well, right there, remember, why do we call them helpers? Because they do what? They help. That's simple, right? They help. So what are they going to probably do with this B cell? They're going to help them, right? And so that's an interaction that we're going to go into a little bit more deeper here, and we're going to continue for the rest of the chapter. So, but let's start out with our friends, the dendritic cells.
The B cells only interact with the costume.
Yeah. Yep. And they're I shouldn't say only. That's their primary job. If a B cell, say, got infected by a virus, it would present MHC class 1, and a cytotoxic T cell could kill it. But, I mean, that's a rarity, right? That's not a primary, that's not part of our usual immune responses. And you got to be careful when you say only, right? because there's probably an exception to that. Whenever you see a true and false statement that's got only in it, if you're talking about biology class, it's very rare that that would be a true statement, right? It's very rare. And if it's true, it's probably not going to stay true. Okay, so for dendritic cells, they get their name from, as you can see, for this cell, they have really long cytoplasmic projections, and this allows them to acquire antigen. Because their primary job is to acquire antigen and present that information to the T cells, right? Whether it be helper T cells or cytotoxic T cells. And as we just pointed out, they're the main activators of this system because they're the main talkers to those naive cells that have never seen antigen before. So unless they're successful, right, we're not going to mount a successful adaptive immune response.
Are they related to the dendrites?
So they're named after them because dendrites have those long, nerve receptors. So the cytoplasmic extensions that they have reminded the scientists of dendrites. So that's, they just look similar, right? That's the only similarity there. And so, yeah. So just like all our white blood cells, they're going to originate from the bone marrow. But these guys are going to travel throughout the body and take up residence in our tissues. So, of course, they're right there on scene, just like macrophages, for when antigens come in. The difference is that the macrophages are really going to concentrate on what? What is their main job? So we've got a cut, right? Bacteria are running into our tissue, eating us up. Macrophages are there. Dendritic cells are there. What mainly are those macrophages going to do? They're going to phagocytose and destroy it, right? That's their main job. They're phagocytic cells. Phagocytosis is their main job. The dendritic cell, on the other hand, he's going to take it in, and he's going to try and destroy it. He doesn't have as much killing power as that macrophage, though, because that's not his main job. His job is to take this information and present it to the T cells, right? His main job is antigen presentation. And in order to do that, of course, he has to have some phagocytotic ability, right? To be able to break apart the pathogen into pieces, to be able to then present it on MHC class 1 or 2, depending on what's going on, or both. So what are the key features of a dendritic cell? The key feature is what he's doing right here, right, in step 3. Presenting antigen to that T cell. How does he do that? He breaks it into pieces, right, and presents it using MHC class 1 or class 2. Which one is it here? 2, because again, he's only one of 3 other cells that can do it. So it's MHC class 2. Other than the fact that it's labeled right here, there's a clue right here in that what cell is he talking to? A CD4 T cell, right? A helper T cell. Remember, these guys can auto-stimulate themselves. We talked about this last time, right? And then they're going to proliferate and differentiate into memory, CD4 helper T cells, and effector helper T cells, which are going to go out and help other cells, right? Release cytokines and communicate with other cells. So dendritic cells themselves must also be activated for maximum effect. What activates them are their toll-like receptors. So remember we talked about these, these are innate receptors that right in our DNA, we come coded with receptors that recognize LPS and lipotectonic acid and flagellin and mannose and these common known patterns that pathogens have, molecular patterns. So these toll-like receptors recognize that, and they're going to drive the activation of that dendritic cell. right? A dendritic cell goes, oh, this is probably bad stuff, right? So then once, if a toll-like receptor is activated, right, if lipopolysaccharide binds to that receptor, that's going to signal that dendritic cell to do even more presentation, right? So he knows that probably what he's engulfing and bringing in is bad news, and he needs to share that information with our T cells. So he upregulates, right? He does more presentation than he was doing previously. And think about this. If he's in the tissues, he's got to travel to where the T cells are in order to do that. So there's large concentrations in your lymph nodes, right, in your spleen, in patches along the mucosa in your intestines, right, those Pryor's patches where there's a lot of B cells and T cells and macrophages and dendritic cells. So depending on where the interaction happened, that dendritic cell, sometimes they're nicknamed the scouts because they're going to be right on the scene, right, but they're always, as soon as they acquire enough information, they're running to try and find T cells, as many as they can, to find who matches what they're interacting with. Right? So again, their main job is antigen presentation to T cells. And there was a video that a student posted one time in the discussion board that you guys are working on right now, and I can't find it. But I'm going to be going through, right, if you guys haven't done that discussion board yet, make sure you do, right, because we're getting to the end. I'm going to be going through and reading and looking at those. Remember, do not post something that someone else has already posted because you won't get credit for it, right? So those of you guys that waited until now to do this assignment, you've got to click through and look at everybody's before you post yours to make sure you don't repeat, right? So hopefully there's a cool one that I've quit posting for you guys because I don't want to steal it from you, right? So I'm sure somebody has already posted it. But there are a couple that usually come up a lot, and I'm going to create a list after I go through and share with everybody. So if you didn't particularly pick somebody's one to reply to and you didn't look at it in detail, there's a couple out there that are really good that I suggest that you take the time to watch. Okay? And so I'll make an Erica's favorites list or something. But there's one I've lost track of. Maybe somebody found it again, I hope, because it was good. It showed the dendritic cell in the tissue traveling to the lymph node. It was an animated video and presenting there. So it's kind of cool. I don't know, some of us seeing it in action helps. So then that brings us to our macrophages. And as I said, really their main job is killing. Right? Although they can present, right? They're one of the professional antigen presenting cells, so they do have MHC class 2. They can talk to helper T cells. They also have MHC class 1. They can talk to cytotoxic T cells. They help in a process that we're going to talk about a little bit later because helper T cells can differentiate into two sets. Th1 and Th2, right? So we number them, set 1 and set 2. And so they produce a different group of cytokines. Because remember, these chemical messages are what are going to drive which way the response goes, right? Which is something we're really going to focus in on next time, which is, a cellular response versus an antibody response or humoral that we haven't really talked that much about yet, right? We've been really focusing on the cellular response right now. We're going to switch gears very soon and talk more about that. So you'll see that in the very last section, which is all B cell and antibodies, we'll really focus in on that particular help. So, wait, what? They had that in there. And I said, I believe that. B cells, they're antigen-presenting cells too. But again, not their main role, right, isn't antigen presentation. But it's necessary for their functionality. Their main role, right, so what's their main roles? Their main role is to produce antibodies, something we've only briefly talked about. In doing that, they need to be able to present antigen via MHC class II to helper T cells to get that help that they need. So you'll notice here, right, a B cell has a B cell receptor. But notice that looks a little different than a T cell receptor. And remember, B cell receptors and T cell receptors bind to antigen. But T cell receptors only bind to antigen that's being presented by a cell, right? Via MHC class I or II. Notice here, this is an antigen all by itself. And this receptor looks a little different, doesn't it? It's got this characteristic Y shape, right? Because unlike the T cell receptor, the B cell receptor has two antigen binding sites. At the tip of each one of these arms, do you see how right here it's interacting with the antigen right here? Another antigen could interact over here. Or depending on how big the antigen is, the whole receptor, both antigen binding sites could bind to it. Not shown in this picture. So, okay, he's recognizing antigen all by itself. That's an important difference between B cells and T cells as applies to their receptors. But then he needs help, right? Because he ran across this bad guy, but he's not quite sure, right, what to do about it. So he internalizes it. He processes it through an endosynthotic pathway, presents it via MHC class II. Right? He is an antigen-presenting cell. He presents the antigens he's come in contact with. And of course, it's MHC class II, so he can only talk to who? What type of T cell? Helper T cell, because the helper T cell has what co-receptor that recognizes MHC class II? CD4 or CD8? This is CD4. So this T cell, right, its receptor also recognizes that antigen. Its co-receptor CD4 recognize the MHC class II. These guys have their secret handshake, their little club they're in, right? And they talk back and forth to each other, right? They each release cytokines, right, confirming this special handshake that they're doing. That's going to activate this B cell in most cases, right? If the T cell is recognizing that same antigen, he is going to cause the activation of that B cell. And remember, whenever we activate lymphocytes, B cells or T cells, we don't want just one of them to go out and fight, right? One's not going to be enough, right? There's millions of bacteria in here maybe, or millions of viruses. So that part of the activation is going right into proliferation, right? Where we're going to make copies, lots of copies, millions of copies of this activated now B cell. Now remember, that B cell, how it knew how to make that receptor that recognizes that antigen is in its DNA. But, something we're going to talk about in the last section, both B cells and T cells, each one of them is made different, right? Keep thinking about this. Each one of the B cell receptors is different for each B cell, right? On its surface, all the ones it makes are all the same, right, for one B cell. But if you then look at his cousin next to him, he makes only one type of antibody, and they're different from his. So again, we're going to proliferate, and we always want, especially with adaptive immune response, what's so awesome about it? Memory cells and effector cells. In this case, the effector cell for a B cell is called a plasma cell. And the reason for that is its cytoplasm gets really huge, because there's one organelle in there that gets really huge. So think about this. If you're going to make proteins, which is what antibodies are, in very large numbers, because they're actually going to make them and secrete them now. So what was on their surface as a receptor, they're going to make it and secrete it outside their cell. So what makes proteins inside your cells? What makes proteins, y'all? Starts with an R. Ribosomes. Oh, we're awake finally. Okay, ribosomes. Okay, and then we have two types of endoplasmic reticulum, right? One we call the rough ER and one we call the smooth ER. Why is the rough one called rough? What makes it rough? Yeah, it has ribosomes embedded in it. And so especially for proteins that we're going to secrete, they're made in the rough ER, right? And they're transported via vesicles to the Golgi apparatus, which is like the packing plant, sends them on their way, in this case, out of the cell. So the rough endoplasmic reticulum, like they have electron micrographs of this, gets enormous in these plasma cells. I mean, just huge. Because we need it, right? They're differentiating to do a pretty important job. They're going to make antibodies to be secreted. Right, so first step is actually binding to the antigen via their antibody. So because that's basically a receptor, right? The B cell, that's the B cell receptor, its antibody on its surface is a receptor. That's receptor-mediated endocytosis. So again, we're going to internalize it, we're going to break it down, then we're going to present it via MHC class 2, which is going to allow them to communicate with helper T cells because they have the co-receptor CD, what? 4, right? And they're going to communicate with each other so that hopefully the B cell is activated if they actually recognize this antigen. We get memory cells, we get now plasma cells that actually secrete antibodies. This is commonly referred to as the humoral or antibody response. So remember when I said, don't say only or all, right? So for most B cells, they need the T cell cytokines to become fully activated. Right? And of course, they can't do this without presenting the antigen they've come in contact with. So that's why they're antigen-presenting cells. So that the antigens that they come in contact via their B cell receptor, they can present that to get help from the helper T cells. So what role do they play in antigen presentation? It's really kind of a selfish role, right? They need help. They need help in confirming that what they've interacted with, how they should respond to it, and if they should. In order for our cells to interact with antigens, the antigens have to have certain characteristics about them, right? And one of the characteristics that tends to be really helpful is if they're large in size, right? So they've got to be big enough that they will actually interact with them, right? Take them in and process them. So what if we want to make a molecule more immunogenic? What does immunogenic mean, by the way? All right, we haven't talked about this yet, actually, because I don't know why I didn't put it in there. Probably because we might save it till antibodies. So the word antigen actually means antibody generator, right? Because back when we were first discovering what was going on, antibodies themselves were pretty well known before we knew a lot of the components of the immune system, because they're very large proteins, and we will secrete them into our blood even. They go into our bloodstream. And so there were these properties about blood that we studied, this human element, right, this humoral element in blood that was very unique and had this ability to bind to antigens. So those antigens, those foreign substances, they found out later too, were actually what were initiating the body to make the antibodies. So the name antigen means antibody generator. But now we use that term to refer to foreign substances that specifically bind to B cell or T cell receptors, right? Lymphocyte receptors. When we say immunogen, we're being more broad, aren't we? We're not being very specific. We're talking about the immune system. Is the only response the immune system can do is antibodies? No, we could have a T cell response, right? We could have an innate immune response, right? Where we actually phagocytize and destroy the pathogen, and maybe that's enough. Maybe the macrophages and the neutrophils can kill it all, and we don't even have to go to adaptive. That is the case sometimes. But what if we want it to go to that next level, right? Is there ways that we can get the system to go a particular way? And the answer is yes. One of the key things is size, but also being able to stimulate the different components of the immune system. So why would we want to do that? Well, in the case of vaccines, right, if we just say, took some bacteria and put them in a blender, right, chopped them up real nice and fine little pieces, and we injected that into you, do you think you'd have a nice, good immune response? Probably not. It's not alive, for one thing. You chopped it up to a million little tiny pieces. And again, it's a whole bunch of pieces, right? So what's the chances the body might actually react? It actually turns out not very good. Because they've tried stuff like this in the past, right? So there are things that they add to vaccines, and this gets real crazy and controversial. Let's stay out of that realm, right? But one of the things that they add are referred to as adjuvants, right? So adjuvants are things that they add to preparations of things that are pathogenic. And this is to help with the immune response. So what are some examples of these? What are they capable of doing in the body? Some of them will cause the pieces of the vaccine to precipitate, to bind with each other, and make it a larger molecule. Because again, we found that small molecules don't really react with the immune system. The immune system itself has to process it and break it into little pieces itself. You can't give it pre-digested stuff. It doesn't deal well with that. One of the other adjuvants that was commonly used is mineral oil. And what mineral oil did was kind of surround the components, and little bits of the components were released over time. Right? So this is kind of like a time-release capsule they were creating by using oil. Because you know, oil and water don't mix well. Right? Oil tends to want to stay together. And so it would kind of trap the components together, but you know, little by little the components would escape. They don't use this in humans though, because there's some problems that they've run into. Another is they've gotten really sophisticated, I guess you could say, in vaccine design, and that they can add components. They can even manufacture DNA components that will activate and look like bacterial or viral DNA to activate the toll-like receptors. Like they've figured out what code will actually interact. But they can even add other things that are similar enough that bind to the toll-like receptors. LPS, not so much, though. That one found to be, you know, too much of a response. So that, I mean, they're really playing around down to the molecular level of trying to add things to vaccines to get a good immunological response in a safe manner as best as possible. And of course, we want a stronger, better response. We want to get memory cells, right? We want a particular response to a particular pathogen, right? For a virus, we want a cellular response, right? Humoral can be beneficial, but we really want a cellular response, right? For bacteria in toxins and stuff like that, we really want an antibody response, right? We really want to push the system in that direction, right? How can we add stuff to, other than the basic information we need of, like, this is what the bad guy looks like, right? This is what the toxin looks like. Please make an antibody against this, okay? In this weakened form, right? We've modified this toxin so that it's not going to do what it normally does, but there's enough information there that you can recognize the bad guy, right? It's like taking away his gun, right? But he still could be bad. So, drawing can be key, as I said. And this is one of the websites that I sent you to, right? Yep, this is the more detailed page. So this was that one I just mentioned, right? We can go into more detail, even, than their cute little picture. And this is another one I want you guys to kind of focus in on as you're preparing for our next lecture, is this drawing, because we just talked about this interaction right here, right? You guys can draw in more here, can't you? Between this helper T cell and this B cell now. And you know how we got from here to here, right? And they're really focusing in on this part and not this interaction between the T cell and the B cell. See if you can sit and draw that out on your own. Right? What's happening here? Why is this T cell standing next to this B cell? What are they doing? Right? And this is an excellent one where, you know, what's going to happen here after this guy's activated? He's going to proliferate. He's going to produce plasma cells. And I love this cartoon because, look, the plasma cell, it's like he's spitting out the antibodies out his mouth. It's a cartoon, right? But, you know, it's cute. Visualization, right, sometimes helps some of us. And then we've got the memory guy. And so those antibodies are going to directly bind to those antigens. And I want to zoom in on this guy for a second and go over a term that we mentioned previously. And so I think it's the overview page I want to go to. The first one I wanted you to read, and then the next one goes into more detail. So we're talking about the bad guys, right? So here they're labeling this a pathogen. And his little crown, you could say here, has the antigen. So what do we know? What did we say an antigen does? It binds to a receptor. Could be a B cell receptor, or if this antigen is taken in and processed by an antigen presenting cell, it could be presented to a T cell receptor. Okay. Antigens themselves, though, can be very large. So we could even flip this, right, and say this whole thing is the antigen. What the antibody or B cell receptor binds to is a smaller portion, right, where the physical binding happens that we're going to look at is a much smaller interaction. And we looked at this in the first PowerPoint that we looked at right here. So imagine this is that little green guy, right? And these little things poking out is his crown. So what name could we give to that cute little crown that would specifically refer to where on the antigen the B cell receptor in this case or a T cell receptor would bind? It's an important term. They're referred to as epitopes. Okay. So as we start to go talk about B cells, right, the B cells bind those B cell receptor, those antibodies bind to really small pieces on an antigen. That really small piece is called an epitope. Right, and they're showing several of them here. So one thing I didn't notice when I had told you guys once before in your book, oh, I thought I was there. There it is. Okay. I had asked you to watch the cell-mediated immunity animation. There are two. There's a 3D one, right? What is the cell-mediated immune response? Do I need to make this bigger? I apologize. I see you squinting. All right. So when you go to the book, right, online, and you go to chapter 20, throughout the chapter there are links to these animations, but the quickest way to get to them is just go to the section that says chapter animations. So the next chapter we're really going to start differentiating out cell-mediated response versus humoral. So again, if you're not a huge reader, but one of these I will probably show in class like I did last time. I'll probably show the humoral one because I've already shown this cell-mediated one. I'll probably show this antibody-mediated one in class and talk about it next time. But all four of these are excellent and have captioning that's correct. So take the time to look through those. Those are really good references.